Assalamualaikum to Sir Azlan. My name is Aqil bin Muhammad Anwar. My metric number is 23DBT20F016. So today I will show you about gram stain simulation. So let's start. First, we need to create the sample. Our sample. So let water dry on slide for 5 minutes. So basically a gram stain is used along with a culture of the material from an infect site to identify the cause of a bacterial infection. The gram stains provides preliminary results on whether bacteria are present and the genotype, such as the shape and whether they are gram positive or gram negative. So in this simulation, um, we will start with the hit the slide. So we need to gently hit the slide 2 or 3 times through the frame. Uh, Drop in and finish hit. So next, we will flood the slide with crystal violet for 1 minute. Wait for 1 minute and we will rinse with H2O. After rinse with H2O, we will flood the slide with iodine for 1 minute. Rinse with H2O again. And we will decolorize with alcohol for 5 to 10 seconds. And we will flood the slide with, we will rinse the slide with H2O first. And we will flood the slide with safranin for 1 minute. And rinse with H2O. And we will view it under microscope. So as the result, as we can see, there is gram negative and gram positive appear. So the pink color is gram negative and the purple color is gram positive. Okay, so for the next experiment is I will do a simulation about catalyst test. So first, we need to know the purpose of catalyst test is a particularly important test used to determine whether a gram-positive cocci is staphylococci or a streptococci. Catalase is an enzyme that converts hydrogen peroxide to water and oxygen gas. So, the step in catalyst test is first we need put a drop H2O2 on each slide. Next, we will isolate the colony using a stick. First, we'll isolate a Staphylococcus aureus. After we pick the isolate colony, we will mix bacteria into H2O2 and watch for bubble. So, if there strong bubbling, it means is a negative, is a positive. So there is immediate strong bubbling, so there is positive. The question is Staphylococcus aureus catalyst positive or negative? So from this experiment, you can see that there is bubble. So the answer is positive. After done the experiment for the Staphylococcus aureus, post the stick into the hazardous waste. Next, we will do for a Streptococcus pyogenes. Isolate isolate the colony and put in the slide. So if there is no bubbles, it means is negative. So the answer is no bubble, so negative. So tip, remember to dispose of the glass slides in the sharps biohazard container. Done for catalyst test. Okay, so the next experiment is coagulase test. So firstly, the purpose of coagulase test is to identify whether an organism produce the exoenzyme coagulase, which cause the fibrin of blood plasma clot. So the first step of coagulase test is we need to drop of water the slide each slide, and we will isolate the colony using stick. Put in the slide, mix the bacteria with water. Dispose of pig, and then we need to add 1 or 2 drops of rabbit plasma. Then we need to rock slide and watch for clumping. So if there is any clumping, that means is positive, and if there is no clumping, that means is negative. So it's tough. Epidermis coagulase positive or negative. So there is no clumping, and that means is negative. Next, we will do for a Staph aureus. Isolate the bacteria first. Mix bacteria with the water. Dispose of pig. Add 1 or 2 drops rabbit plasma. Rock slide and watch for clumping. So, so if there is clumping, that means that is positive. Wait until 15 seconds. Staph and aureus coagulase positive or negative. So there is clumping, that means is positive. done for stage. Okay, for the last experiment is oxidase test. So the purpose of oxidase test is used to identify bacteria that produce cytochrome C oxidase, an enzyme of the bacterial electron transport chain. Note all bacteria that are oxidase positive are aerobic and can use oxygen as a terminal electron acceptor in respiration. So, the first step of oxidase test is we need to isolate uh, the colony with a sterile toothpick, put in the dry slide. So, if there is a color change, that means it's positive. Dispose the toothpick, pick a new colony, isolate new colony in the dry slide. So, is this one oxidase positive? If there is no color changes, that means it's negative. Dispose the toothpick, pick a new, isolate a new colony, put in the dry slide. So if there is no color changes, that means it's negative. Right? There is color changes, that means it's positive. So finally, we need to dispose dry slide by hazardous waste.